Hello?
Hello, sir.
Sir,
Just
just testing, sir. Actually,
just a minute. I'm trying to connect to my file. I just can't do it. Just a minute.
okay. You can share, sir.
Yeah, my blocked or something.
Okay.
Share is getting blocked. I don't know why.
Sharing problem.
చిన్ను
Hello? రావాలి. Sound up here.
కాదు కానీ ఇది చూడు మ్యూట్లో ఉన్నది.
అందరూ మ్యూట్ చేయండి.
చేసుకో.
Sir, are you able to share, sir? Okay. Now slides are visible, sir. Subba, sir. Sir, your slides are now visible, sir. Give me 2 minutes time, sir. Then we will set the speaker and everything. Then we will start the program, sir. హలో వస్తుంది అక్కడ కదా ఇక్కడ దాంట్లో ఏమైనా పడతారు. Sir, Subba sir, once please respond, sir, whether sound is proper.
Yeah, I can hear you. Yeah, I can hear you.
Yes, sir. Yes, sir. Actually, we are setting sound system, sir, for
Just
more sound, but it's not actually.
Can you hear me?
Yes, sir. Yes, sir.
Okay. You tell me when I to start. Yeah.
Okay. Okay. Sure, sir. Sure. I will. Hello. Okay. Hello.
Hello, sir.
Sir, good afternoon, sir.
Good afternoon.
Sir, please make full screen, sir. Actually, this
Yeah.
presentation mode. Before you starting, I will just introduce, sir. Then we will start,
Yeah,
sir.
sure. Okay.
The students, today we have eminent resource person from Jawaharlal Nehru University, Delhi, and that is none other than Professor Naidu Subba sir. And he studied his MS and PhD in IIT Kanpur, and then he worked in several places, and now he is currently working as a professor in JNU, New Delhi. So today, sir is accepting to deliver a talk on bioinformatics to all your students. So thank you for accepting our invitation, sir.
Thank you,
Thank you very much, sir.
Thank
And
you.
kindly start your session, sir. Thank you.
Thank you. First of all, let me tell you that I'll be just giving one lecture in 1 hour. Then I will take some break, 5 minutes. Then we'll go to second lecture. First lecture, generally, I'll cover mainly genomes and RNA related stuff. Second lecture will be mainly related to the proteins, structural mathematics, as well as little bit structure-based drug designing, which we have been doing. Besides, I also tell you what are the drug targets that can be used for as a resource, kind of a thing. So, first few slides will be mainly related to biology. All of you must be knowing it, but still I'll go through that. Whenever you talk about life, living organisms, they can grow and divide they can produce offspring, they can adapt to the different environments, they evolve to new species. That's why cell is a unit of life. There are three branches of life. One is prokaryotics and eukaryotics. Isn't that there is
Sir,
a
Subba sir,
archaea?
sorry for disturbing, sir.
Yes, sir.
Please put in slide share mode, sir. Was PPT.
Slide share में तो है ना. Next slide is not coming up. Second slide is not coming up.
Hello.
Hello. Second slide is not showing up.
It is only in not in presentation mode, sir. Actually, that is
Full screen में अपने काम
slideshow
है.
mode.
Yeah, I mean full screen. I'm showing full screen only, no? No?
No, sir. No.
Just a minute. Can you see the slide?
No, sir.
Hello? Can you see the slide?
Sir,
Hello?
F7, you please F7.
Yeah, I'm full screen mode in my computer. I don't know whether
Okay.
I'm able to.
Sir, otherwise you please. It is only in just PPT mode only, sir, not in slideshow mode.
Okay, can you see that?
Yes, sir. Now it is okay, sir.
Why it's not showing up? Shall I go to just see that?
Yes, sir.
Can you see now? No?
Now it is same F7, sir. Full screen mode, please. F7. F7.
Yeah, full screen only. I am in full screen mode only. Just a minute. Not full screen. Can't you see now?
No, sir. That is previous like previous only.
Okay, just come back.
Current slide.
Okay. I'll go this way only.
From current.
From current slide or otherwise.
It is okay,
Can you
sir.
go
You
to?
can.
Otherwise this one
There
Oh,
is
just
a circle like arrow mark is there, na, sir. There is
and
full screen.
left side top of the window.
Yeah, that I'm going that way.
Save button. There is some two arrows, opposite arrows, undo arrows. Then there is one full screen option is there, sir.
Yeah, here I'm going to have full screen. I don't know. I am not showing up.
That one, sir.
Yeah, it's okay now? No?
Okay, sir. No problem, sir. You can carry on like this only. We are seeing or just in PPT mode on this.
I just can't see. Wait, what is the problem? I'll again skip it and share it.
Okay, sir.
Can you see now? Full screen?
No, sir. Now the earlier version only.
Why it's coming like that? I can't understand. I have to put entire screen or what? Just let me put entire screen then. No. What's the problem? I can't understand. I just. Can you see now full screen?
Yes, sir. Now full screen.
Hello?
Yes, sir. Now okay, sir. Now
Hello?
full screen.
Huh?
Yes, sir. Now okay.
Second slide?
No, slides are not open, sir. Just only the desktop is seeing.
Oh, shit. Something wrong is going on. I don't know what is it. Can you see now?
Yes, sir. Yes, sir. Now okay.
Huh? Full
Now full.
screen?
Yes, sir. Now slides are is coming. Yeah.
Okay.
You please carry, sir. Most of the students are in Telugu medium also. You can speak Telugu also, no problem, sir.
Yeah, a little bit. Yeah. I'm used to
Yes, sir.
English only. Anyway, mm, uh, first of all, let me tell you that living organisms, they can grow and divide. Uh, they can also produce an offspring. They can adapt to the different environment and they evolve to new species over the years. Uh, and there are three branches of life. One is archaea, bacteria, and eukaryotic. Archaea and bacteria, we call them prokaryotic system organisms, and multicellular organisms, we call eukaryotes. And these are how the evolutionary tree of the three kingdoms. And again, you see that archaea, bacteria, they form prokaryotic and eukaryotic species, fungi, plants, and animal cells are there. And this is the prokaryotic cell where nucleus is not enclosed, and cytosol, cell wall, and everything is ribosomes, everything is nearby the cytosol. Whereas plant cell or animal cells, there are different organelles. Each organelle functions have different roles, and nucleolus synthesis of ribosomes. And these are varieties of organelles. All of you heard of it in a given cell, mitochondria and all. Mitochondria has got its own genome compared to the nucleus where it has got the genome. Uh, there are four organic macromolecules or what you call polymers that are made from simple building blocks, what you call carbohydrates, lipids, proteins, and nucleic acids. This class I'm going to talk about nucleic acids. Next class I'll talk about proteins. When you talk about nucleotides in DNA, uh, there are many species have got different chromosomes. Fruit fly has got 8, and amoeba has got 13. Human has got 46 chromosomes, 23 pairs of chromosomes. And that way, there are particularly adders, strong parent has got 1200 chromosomes. That way, there are number of chromosomes vary. Each one is a prokaryotes is circular, but eukaryotes is a linear chromosomes. And in a human, the genomic hierarchy in eukaryotes is nuclear genome, and there are 23 pairs of chromosomes, and each chromosome will got two DNA molecules, uh, and each genome, human genome has got 20 to 21,000 genes in the human genome. It contains around 3 to 9 bases, nucleotide bases in the genome. So these are the what you call nucleotides, nucleosides, and purines and pyrimidines. Again, adenine, guanine is a purine, and cytosine, thymine, and uracil is a pyrimidines. And again, nucleoside when you add the ribose group, and then you monophosphatide you add it to become the nucleotide. And then depending on the uracil or thymine, they call RNA or DNA deoxyribonucleic acid or ribonucleic acid. We call generally DNA is a double stranded, RNA is generally single stranded, particularly messenger RNA. And we'll talk about it more detail. And these are the individual nucleotides, the structures. All of you might have seen that. These are called bases. And these are the pyrinucleotides, ribose as well as the phosphate group, which are the backbone of the DNA. And again, it has got a sugar moiety, ribose, deoxyribose. Uh, in DNA, there are two strands, whereas RNA is a single strand. Uh, in DNA, it is thymine, and RNA is uracil as the nucleotide, which differentiate between the RNA and DNA. And This is a deoxyribonuclease and ribose nuclease. OH group, which is can be attacked by other groups. That's why the RNAs are more unstable than compared to the DNA. DNA is a double stranded, that's also more stable. That way, one can generate DNA structure, but you cannot solve the whole DNA structure. Even nucleosome of the DNA is not solved very well. And this is the double stranded DNA, and this is a single stranded RNA, and how the nucleotides add up, making the double stranded, single stranded DNA and RNA. And as I told you, the genetic information is stored in the deoxyribonucleic acid. In some viruses, RNA also can become a storage, a genetic storage information. DNA contains all the information needed to build on the individual, particularly genome, all the chromosomes together, the complete genome, nucleate, all the chromosomes together, whatever the nucleotides that they have, they the genome has got all the information that can be what one organism is going to be after adapting to the environment. So the entire complement of genetic material carried by an individual is called genome. Eukaryotic cells usually have more than one genome. Uh, Human, nuclear, and mitochondrial. Nuclear DNA we are talking about. Mitochondrial generally uses for generating energy. It is generally carried from mother to children. Some eukaryotes have a single genome. It has got some have four or more genomes. DNA makes copies of itself. DNA encodes information. DNA changes by mutation due to certain carcinogens, radiations, and things like that. And DNA can get replicated using different varieties of enzymes, and these are the enzymes listed here, and what is the function in the replication process of the DNA. When the cell grows and divides, then it produces its daughter cell, exactly same copy, and the cell divide, and then DNA get replicated. Within the cell, the chromosome structure looks like this one. This is a when it gets condensed DNA double stick helical structure, then there are histones form a there are four histones uh, the core. Then the DNA gets wrapped onto the histone molecules, and this is what you call a nucleosome. And nucleosomes uh, form a coils and then supercoils, and it becomes a chromosome, which very highly condensed. That's why whole chromosomes very so condensed such a way that it occupies very less space. This DNA strands wrap around the histone protein, forming a nucleosomes. Nucleosomes coil together, forming a chromatin. Chromatin loops and coils together, form supercoils, and supercoils bunch together, forming a chromosomes. So this is what it looks like. All the histone molecules, histone 1, 2, 2A, 2B, and histone 3 and 4, and the 108 residues wrap around the histone tetramer, and one can go to the this particular site and see the video of the nucleosome structure. And DNA has got varieties of conformation like bDNA, aDNA, zDNA, and four standard DNA, particular telemeric. Every chromosome at the ends there's a telemeric DNA. There's a quadra nucleotide, this thing rather than double stranded. And the varieties of conformations of DNA, different segments have got different conformations. DNA cannot be solved. Old chromosome structure cannot be solved. So it's so condensed. Only fragments of DNA and their forming complex with the from proteins can be solved. That's why there are fragments of DNA and their corresponding binding sites with the certain proteins made the complex, and people have solved the structures. That's why complete DNA can be generated as a double helix, uh, major groove, minor groove, and all, but you cannot solve the structure. This is again when DNA to RNA, RNA to protein. That's what when nucleosomes they relaxed on the this thing, then the certain part is exposed. Then the RNA polymerase and the different transcriptor binds in the promoter region and regulate particular gene and transcribe into that particular gene to mRNA. That mRNA get translated into the from the ribosomes and produce the protein peptides. and then protein peptides form a certain fold and with tertiary structure. That is why for many years biology was not very quantitative. Uh, 1980 onwards, people have started developing the database of gene sequences, protein sequences. First, the database was the protein sequence database. Uh, by day of. Then the nucleotide sequence developed. All the people have started putting into the database ESTs and complete gene sequences from the different organisms. Later on, it became a gen bank. That's why in the research in biology, from the experimental point of view, there people have generated different genome sequencing of the chromosomes of a particular organism and the nucleotide sequence 5' to 3', and all the nucleotides in what order they are occurring. A complete genome is sequenced for all the chromosomes and put into the chromosome nucleotide sequence. From that, once you get a chromosome sequence, then they try to identify the genes that are in the particular DNA sequence of the chromosome, uh, nucleotide sequence. From that, there are many programs are there for gene identification. I'll go through later. That is why later on, then try to find out what gene, what kind of translation to mRNA, how it gets translated using a certain genetic code. That is why mRNA to protein, there is a genetic code which is very well understood. Sequence to structure also there is a code, but we don't understand at the moment. Protein-protein interaction, protein-ligand interaction, there multiplication, there is a code, but we don't understand at the moment. So by 1918 onwards, bioinformatics era started. Since computers have come up in 1993 onwards, browsers have come. What are the program they make it, program or database they develop, they make it available online so that one can give input and get the output from the an input certain program runs on the background. Similarly, certain database they develop it, make it available to the on the website so that they can access those resources publicly to different if you've got a good internet connected to you and all. So that's why 1995 onwards, human genome sequencing, other organism sequencing has been done. That's why it become a bioinformatics era started with what you call fourth genomic era. Uh, Bioinformatics has become very important, is information intensive science and what you call in silico approach. There are varieties of definitions are there. The Bioinformatics Committee, they defined bioinformatics as a research development or application of computational tools and approaches for expanding the use of biological, medical, behavioral, and health data, including those of acquire, store, organize, archive, analyze, or visualize such data. So the biological data, which is coming up in large scale, environment tools have been developed, and then they did a lot of research to identify the genes function, what kind of gene expression studies nowadays, RNA sequencing, next generation RNA sequencing. So much of data is coming for the last 15, 20 years, particularly next generation sequencing. Whereas computational biology, they define as the development application of data analytical and theoretical methods, mathematical modeling, and computational simulation techniques to the study of biological, behavioral, and social systems. These are the well-known definitions for the bioinformatics and computational biology. And there are other definitions are there. Uh, Different institutes, different scientists have defined these definitions. When you talk about from DNA to genome, uh, Watson Crick, 1953, solved the DNA as a double helix, and AT and GC make a pairing by hydrogen bonding. Then they made a from the crystallograph data from one of the scientific group. They got the data about the AT-GC hydrogen bonding pattern. Then they're able to make the model, DNA model, and then they explain how the DNA gets connected, uh, map double stranded DNA, and then they got the information. Then Sanger started sequencing the proteins and get the amino acids one by one, and sequence alignment programs. Uh, uh, because of internet access later on, many databases have come. Later on, Sanger deoxy DNA sequencing expedited the sequencing. Then PCR polymerase helped in getting the RNA sequencing much more quickly. Nowadays, people do RNA sequencing rather than the genome sequencing. Genome sequence as a reference, they can find out which genes are expressed in a given cell, and they can find out map to the where the expressed genes are coming from, which region of the genome. Some protein sequences have been most of the protein sequences that are evolved are from the gene sequences. I'll tell you what is gene structure and all, and prokaryotic and eukaryotic, and from there they identified the exonic regions of the gene sequence. They are assembled and they get mRNA sequence. They translate to protein sequences, and some proteins which are isolated, they solve the protein structures. Hemoglobin, myoglobin, and lysosome were solved in 1965 onwards. Later on, for last 40 years, more than 2 lakh structures have been solved and deposited in the protein data bank. GenBank started in 1983 or 84, and there are many uh, CISPROT and NCBI, National Center for Biotechnology Information. Uh, there are varieties of programs like FASTA, BLAST, World Wide Web. Each genome has been sequenced. Then the Human Genome Initiative by 2000, first genome draft has come. Then many other bacterial genomes have been sequenced by that time. And then they started many genome sequencing centers where the different organisms, they started sequencing the different genomes of different organisms. Once you get the genome, they identified the functional genomics research centers where they annotate the genomes, finding the genes and what function, what kind of genes, what kind of proteins, what kind of mRNA code, and what kind of proteins they produce, and what kind of function they'll have by gene ontology kind of technology they tried it. Proteomics is more difficult one to in a given cell to isolate the proteins, separate them, characterize them is very difficult. Protein sequence is the most difficult one because you have to get the fragments of protein and then each fragment mass spectroscopy, and then they find out the molecular weight. Then they find out which amino acids of fragment can combine to give that molecular weight, and then they identify the sequence particular fragments, and then they assemble the fragments to a protein sequence. So this is more proteomics is more difficult to get the protein sequence, whereas that's why most of the protein sequences that are available in the gel peptide or CISPank, CISPROT or TRMBL mainly annotated from the gene sequence identifications and assembling the exons and then from the gene sequences. Then people started the structural genomics centers to start isolate the proteins, okay, make a crystals, then solve the structure by X-ray. Nowadays, some small proteins like 200 amino acid protein they can solve by NMR. Some proteins can be solved by membrane proteins can be solved by electron microscopy, 5 to 8 resolution structures. Nowadays, we talk about system biology, where we simulate the cell biological processes in the cell, metabolic pathways or signaling pathways, and can simulate, understand the different this thing. When you talk about bioinformatics, there is Genome Online database which provides you all the genomes that are being sequenced. There are 5,52,000 genomes that are being sequenced for more than 5 lakh organisms, and 36,022 completely sequenced, which contains archaea, bacteria, and eukaryotics. 4 lakh genomes, there's permanent jobs have been developed, and 1 lakh 15,000 still they're sequencing, and hopefully they'll be completing very soon. Most of the gene sequences that are deposited earlier by individual partial sequences are what you call ESTs, or express sequence tags, uh, so from the exonic regions. They deposit in GenBank. That's why GenBank has got many divisions. It's got almost 37 million, 371 million protein sequences, and this many number of bases. Then individual genes, mRNAs, ESTs, STS. Nowadays, genome sequence, whole genome of the organism also deposited as a one entry in the GenBank. This is the 2023 December release of the GenBank. CISPROT at the moment are like 5,70,000 protein sequences. Most of the proteins in CISPROT are well characterized. Function has been annotated by individual scientists who study that protein for a long time. whereas TRML almost 250 million protein sequences are there, which are mainly translate from the mRNA sequence to the protein sequences in the genetic code. And PDB has got 2 lakh proteins, 183,000 X-ray, 14,000 from NMR, around 19,000 from electron microscopy. Electron microscopy structures are 5 to 8 angstrom resolution, where NMR X-ray structures are more accurate, more less than 3 angstrom resolution. There are many chemoinformatic databases which are publicly available, PubChem, ChemBank, NCI, Maybridge. Many chemical and pharma companies, since there's a large number of molecules, they procure them and test them for high throughput screening to identify the active compounds in a given target or cell lines. So these are varieties of small molecules are available in these databases. So there are varieties of biomolecular databases like sequence structural databases, protein sequence database, DNA sequence databases, 3D structure database, structural motifs database, sequence motif databases, genome sequences, annotations, individual genome, what are genes, promote regions, stop codons, and then the splice sites. Then each gene can code more than one protein in different cells to what you call isoform due to alternate splicing. Then the comparing the genomes, find out functions. Then they do molecular function assignment, transcription regulation, all the transcript factors, what you call regular and interact, this thing, and then all the enzymes, pathways. Then you do study the metabolic pathways like signaling pathways, protein-protein interaction, and gene regulation pathways. So if you go to vls3d.com, you'll find all the computational tools that are available for bioinformatics, all the databases that are available publicly online. They make it available on the VLS3D. There are links are available. Whichever database you want to go, whatever the program you want to run it and give input and get the output, there all the tools are available in VLS3D. It also has got a large number of tools for structure-based drug designing, uh, or virtual screening, kind of databases. Bioinformatics as a chemoinformatics tools are available in this particular VLS3.com, which is a comprehensive website for all the tools, databases, as well as the structure-based drug designing tools, particularly QSR, QSPR, or ligand-based drug design like pharmacophore modeling, as well as all the tools are available here. So if you come to biological databases, what you call primary database like GenBank, Sysport, and TRML, we call it primary databases. From that, the people have derived many other databases we call non-redundant database like WOL, SP plus TRML together composite database. Then there are from the primary database, they derived the what you call ProSet patterns, which are functionally important residues in a given binding site, which has certain pattern. Same pattern will be there in many sequences having a similar function. Prints is a many multiple element of proteins. There are certain regions which are consensus conserved regions are available. They're called fingerprints, and each signature, there are different signatures are there in a given family. Some protein may be having 5 signatures, one may be 4. That way, there are consensus signatures, which are called fingerprints. And profile is a consensus sequence along the with respect to all the 20 amino acids each position, or the relative occurrence of the amino acid that can occur, and protein families, blocks. When you do multiple amino certain consensus regions are there, and from that you can make a blocks database, mutation database, and identity database. And then there are interpro databases, a integrated protein database. Given a protein, you can find out all the individual domains which can function, structural domain, functional domains in a given protein sequence. What are which family it belongs? What are the binding site residues? If some already known other protein. So these are varieties of information available from these biological databases. So nucleotide sequence database are mainly GenBank, EMBL, and DDBJ. Is a consortium of 3 organizations. They make the sequences. Sequences, even nucleotide sequence is the same, but the entry information that they provide in different fields of the particular gene sequence, and they're different. Each one provides a different kind of information in their own format. So this is GenBank has got 12 traditional divisions: primates, plant and fungal, bacterial and archaeal. That way, there are 12 traditional divisions, and there are 6 bulk divisions, what you call EST, genome survey sequences, high throughput genomic sequence, STS sequence tag site, high throughput cDNA and patent kind of thing. So from the biological data, what you call DNA sequence, you can go to the mRNA sequence, gene expression data through microarray as well as nowadays RNA sequencing data. Then once you get the RNA sequence, then mRNA sequence is known, so you can translate to protein sequences. From the protein sequence, one can generate a model of the protein, or somebody can isolate the protein, crystallize them, and solve the structures. And if you know set of proteins which are in a certain biological process, are they are correlatedly expression in the gene expression, and then see which process is active. Then a certain pathway is active in a certain disease condition compared to non-disease cells. Then you can say those proteins in the particular process can be a drug target. And then there are varieties of biological images one can make from the uh, different diagnosis and other studies. GenBank sequence entry will be like this one, locus and definition, then the access, and finally this is the entry. And here they give you the DNA sequence. There are large number of fields are there, and what are other sequences similar to that sequence, orthologous, paralogous sequences that are there, and this gives you the protein sequence. And again, they give you different segments, promoter region, regulatory regions, and the corresponding protein sequence, the DNA sequence. This EMBL format. There are varieties of formats are there. One can convert one format to another format by read sequence program. And there is a Entrance is a browser which integrates different databases like PubMed, Nucleotide Sequence Database, OMMI, online Mendelian inheritance of man, uh, gene disorders database, and genetic maps and genomes, protein sequences, and each sequence the structure is available, link it to the structure, and any journal related to article related to the nucleotide sequence or protein sequence or protein structure, they link it to the different literature database as well. So that Entrance is a very good browser integrating all the database in the this thing. This is a gene structure of prokaryotic, and there's a regular region promoter, then the terminate region, and there's a one transcript which can be spliced into different transcripts, and that way you can produce major transcript can be cut into different proteins by cutting it, and that way eukaryotic there is no introns, whereas eukaryotic there are many introns and exons. Uh, and exons and introns, there is what you call as splice sites, which sometimes what happens due to splice sites, there can be alternate splicing due to which different combination of exons can form different protein sequences, what we call isoforms from the single gene. And there are different core promoter, proximal promoter, then the uh, activator regulators regions are there, suppression regions where some molecule can bind, gene regulation can be stopped or